Okay, so I think thanks a lot for the introduction. And it's first thing to do is to actually thank Ian for the opportunity and the organisers to speak today. And as Ian said, there's a lot of people that have worked for a lot of years in the antibody engineering field, and actually for me personally, coming back to this meeting, which I haven't been to for a few years now, it's been really great to see so many old friends and collaborators and pick up on some of those discussions. And I think Ian's right that a lot of what we do is about those connections and collaboration. So thanks for the opportunity. Sort of continuing Ian's Nativity Christmas theme, I must admit I feel a bit like a character from Dickens's Christmas Carol. So I think I'm up first as the ghost of Christmas past to talk about making HumiRa, which, as Ian said, did start a long time ago, back in the early 90s. So a lot of the work that you're going to see this morning is very much a retrospective. There's some quite probably relatively basic science compared to some of the fantastic stories that we've been hearing about over the past couple of days. But hopefully you'll find it an interesting, nice story, and I'll try and put it in a little bit into context around where we are in terms of treatment of rheumatoid arthritis today as well. So as Ian said, the title is How Phage Selection Produced the World's Biggest Selling Drug, and I will touch a little bit more on phage and the balance of technologies. Sort of slight disclaimer to say, so in 2012, worldwide sales for HumiRa were 9.3 billion. It was the largest selling biologic. I have to admit, I think it might slightly have been pipped by Lipitor in terms of the overall world best-selling drug, so I'll get that disclaimer out. But it's predicted to be for 2013. And as you all know, it was originally approved for rheumatoid arthritis. It's now approved in many other indications in the US and also in Europe. So what I will do is a little bit of scene setting around the disease, rheumatoid arthritis, and where the biology of tumour necrosis factor alpha was back in the early 90s, and I think it's sort of quite important to put a little bit of historical context on that. I'll then walk through the discovery process and explain the contribution that we at CAT made to discovering the molecule that became HumiRa. A couple of slides on rheumatoid arthritis today, because I do think rheumatologists have been real pioneers of adapting and bringing on board biologics treatments, and I think that's a nice story. And then just a couple of slides talking about the balance of phage and transgenic mice and what else might be to come. So pretty much everybody knows about rheumatoid arthritis, the disease and its impact. It is commonly associated with joint pain from aging, from injury. It's progressive, it's disabling. It really does impact the quality of life of patients. And I think that's really important to focus on, that the inflammation generally leads to joint damage, bone erosion, cartilage erosion, tends to affect the small joints in the hands and feet, so it is disabling, but it can affect any synovial joints. And not to labour the point too much, but this joint erosion, this severe inflammation, does really result in a lot of chronic pain, so patients feel generally very unwell and fatigued. It's a pretty prevalent disease. I think it's somewhere around 0.5 to 0.1% of the adult population, so quite significant numbers of patients worldwide. I think around about 25 million worldwide, really significant patient burden. So we started working on looking at developing HumiRa back in the early 90s, and I think it's worth just pointing out a couple of the key contributions in terms of the biology of TNF alpha that were made in those early days. And it's always hard to sort of pull out contributions, but I do think the work that George Collias did in terms of setting up preclinical models of arthritis in transgenic mice were pretty pivotal at this point. So he published a paper in 1991 which reported a transgenic mouse with human TNF alpha, which had a lot of the phenotype of rheumatoid arthritis, a polyarthritic mouse. And he actually went on through a number of papers with collaborators looking at how inhibiting TNF alpha could actually help reverse this phenotype. This is actually a slightly more recent paper. So a really strong clinical model was in place, and that was translated really into the clinic by Mark Feldman and Ravinder Maini, who won the Lasker Prize in 2003 for their clinical work on tumor necrosis alpha and how that got translated into treating rheumatoid arthritis patients. So we're talking 1992, 1993. They started work on clinical trials actually using infliximab, which is the, as I'm sure you know, the chimeric anti-TNF alpha, which was made by Centocor, and they started seeing some quite promising clinical results. So we got to a place where we had some early phase 2 data for infliximab and also for Enbrel in rheumatoid arthritis, and this really started pointing to the fact that tumor necrosis alpha was pivotal in the cytokine inflammatory cascades in rheumatoid arthritis. So it was really thought that TNF was triggering the production of a range of different pro-inflammatory cytokines that caused inflammatory cell migration into the synovial joints, such cytokines as IL-6, IL-8, GM-CSF receptor, and also IL-1, which was implicated in the actual bone destruction and cartilage destruction. So we had started to build a picture of TNF alpha biology with quite a nice disease linkage. So that's just a bit of context in terms of where the biology was. So what did that mean in terms of getting started on this journey of HumiRa discovery? So worth giving a bit of context on where we were in terms of Cambridge antibody technology. So this is a very nostalgic picture of the labs that we moved into in 1992. So this was just about the time that I joined CAT. We just moved from the Babraham Science Park in Cambridge to this building called Beach House down in South Cambridge in a little village called Melbourne. And the letting agents were really excited about the idea that this was going to be a great biotech hub. So we had this one lab, which was downstairs. Upstairs, we had a coffee room and a photocopier and a kind of pretend reception area to make us look like a proper company. And it was a really exciting time. And this is the first staff, one of our first staff photos. So this is the entire staff of CAT in 1993 in front of Beach House. As you can see, we weren't chosen for our sartorial elegance. We were quite an interesting set of people, but quite dedicated. And I think Dave Chiswell, who was our CEO at the time, must have taken this photo because he's not actually on it. So we were a small biotech, a couple of dozen staff, and we started having some conversations with Knoll Pharma, who were Knoll BASF actually, back in late 1992. And these conversations resulted in discussions around a potential agreement, which was our first significant business deal. And again, just to keep reminding you of the context, so human antibodies certainly hadn't fulfilled their promise at this stage. I mean, Ian gave a very nice summary of where we were in terms of the technologies in the early 90s. So, I mean, OKT3 had been approved back in 86, which was the first murine approval. We hadn't had a chimeric approval yet. The first chimeric approval was ReaPro, which was 1994. So still early days, but therapeutic antibodies were certainly an exciting area to be working on. And phage display, as Ian very nicely explained, was still in its infancy. So we were working very hard on building large, naive libraries that weren't that large actually at that point. So we were learning the technology, we were learning how to do the cloning, and we were learning how to do the selections. But on the back of that, we saw, and certainly our colleagues in BASF saw the potential for this opportunity in differentiating from Remicade and Enbrel in rheumatoid arthritis. And what they brought to the table was a lot of interest and expertise in the TNF alpha pathways, and they had a very valuable reagent which was called MAC195, which was an anti-TNF alpha murine antibody that they had actually taken into phase 3 for toxic shock at this point. So quite well validated antibody. So we went and started building some fairly basic design goals at this point for the creation of adalimumab. Pretty simple, so it had to be specific, it had to bind TNF alpha, it had to be of high affinity, at least the same affinity as MAC195. And then we started to think about competition and differentiation. We were hoping to achieve a 2-week half-life so that we could have a dosing regime of every other week. And obviously we wanted to make this antibody as human as possible to reduce any of the risks of immunogenicity so that we could use it as a sustainable treatment over a long period and potentially either use in monotherapy or in combination. So this is an overview of the basic discovery process that we use, starting with this mouse antibody, MAC195. We made a human version of that using a process called guided selection, which I'll explain in a minute. And then we were left with quite a challenge in terms of increasing the affinity and potency of that antibody to take it up to what we needed in terms of efficacy. So I'll talk through the guided selection piece, then I'll talk through the optimization. And as I'm sure, well, hopefully you remember and are aware, the process of guided selection was one where we took the mouse monoclonal MAC195, cloned that into single-chain FV format so that we could then use that to plug into our phage display vectors, which were all in single-chain FV format, and pair that with human either heavy or light chains. So you start with the mouse single-chain. You keep the mouse VH region constant and pair that with a repertoire of human light chains, select on TNF alpha, and end up with a chimeric mouse-human VHVL. And then you go through the process again, but you keep the human chain constant. So you then go and pair with a new repertoire of human heavy chains to hopefully end up with a fully human single-chain FV. So this was the theory. Worth just explaining why we chose guided selection, because I guess at that point the obvious thing to do would have been to have CDR grafted MAC195. And the reasons we chose the guided selection was, well, first off, we wanted, as I said, to make this antibody as fully human as possible, so we didn't want any rodent residues retained. We wanted to preserve the original epitope. Obviously, you could have done that using CDR grafting. But we felt that the phage format would give us quite a bit of flexibility in terms of optimizing the antibody. So we were starting to learn how you could make mutagenized libraries and increase affinity. So we actually felt that was quite an opportunity for us. And we also thought that could eventually generate more than one lead so that we would have more choice in terms of what we took forward for the next phase of development. So it was a very active decision to go down the phage route and use guided selection. And we went through this process of conversion, pairing the MAC195VH with our light chain repertoire, which was a massive repertoire of 8 times 10 to the 5 in those days, but that gave us enough of a variation to be able to select a chimeric heavy and light combination, and then we paired that with the human heavy chains to give us this clone called 4SE3, which had a KD of 100 nanomolar, so not great, but it was specific. It competed with MAC195, and MAC195 at a KD of 0.6 nanomolar. So having managed to create a human version, albeit of lower affinity, we then went on to work on optimizing the properties of the antibody to increase the kinetic characteristics to enable us to deliver the original design goals. And this was really done in 2 phases. So the first thing we did was we actually repeated the light chain shuffling to try and find a light chain partner that could potentially be of higher affinity. That was reasonably successful, and we came out with a clone called 2SD4, which had quite a similar light chain to the parental one, but with a few point mutations. And then we went into a fairly rigorous optimization phase, where we mutated the heavy and light chain CDR3s quite substantially, and we also did a 10% mutagenic spiking of the other 4 CDR1, 2 of the heavy and light chain. And we did a lot of selections based on reducing the concentration of TNF alpha and a lot of long off-rate selections. And we ended up using a Beercore off-rate screen to try and find our improved variants. So this is just it's actually a screenshot from Tris Vaughan's lab book summarising some of those data. So the original clone that we were working from was 1.4 times 10 to the minus 2, seconds to the minus 1 in terms of off rate. And we managed to identify a couple of clones, heavy and light chain, which had quite significantly improved off rates. And the really reassuring thing that we found when we actually put those back into single-chain FE format and did some actual in vitro cell-based assays to look for function, we did see that improvement in off-rate actually correlated with an improvement in potency, which was something that we were very keen to demonstrate. So in those days we used radiolabeled iodines. This is radiolabeled iodine, TNF alpha, and just looking at inhibition of binding to U937 cells. So I think this is the original MAC195, and this is the improved variant. So just to summarise in terms of that process, I sort of talk through the fact that we started with the MAC195, worked down to a human clone that we did a lot of work in terms of mutagenesis to create a clone called D2E7, which was then reformatted into IgG1 to give us D2E7 in whole antibody format, and that had a KD of 0.09 nanomolar, so it actually was significantly improved over MAC195. So, and just to demonstrate the number of changes that we were producing through the optimization phase, there were around about a dozen changes in the heavy chain compared to the original human starting clone, and around about half a dozen in the light chain. So not too many mutations. So this clone became adalimumab, the MuMab at the end, obviously reflecting the fact this was a fully human clone. The brand name is HumiRa, which is human antibody in rheumatoid arthritis. Anyone who's ever done an ELISA will know why we called the clone D2E7, which was the clinical phase clone. And just to, again, a bit of nostalgia, these are actually screenshots from, again, Tris Vaughan's lab book, just showing the original isolation of the heavy chain clone, which was D2. So, and this tells you this was isolated on the 24th of February 1995, and the light chain was actually isolated a few days before E7. And these were solution phase selections with very long off rates. We had a tradition of giving pet names to our favourite antibodies back in the early days of CAT, and D2E7 also became known as Eric in the research phase. And Eric was actually named after Eric Cantona, and I don't know how many of the people in the room remember Eric Cantona, but he was signed to Manchester United in the early 90s and was one of the most prolific strikers, fantastic. We used to follow a lot of football in the labs in those days. And he also had a slightly philosophical streak, and he was actually well, he was actually suspended for 8 months for drop-kicking a Crystal Palace fan who gave him a bit of lip during a Premier game. And when interviewed about this, he very philosophically and enigmatically came out with this very famous quote of, "When seagulls follow the trawler, it is because they think sardines will be thrown into the sea," which was quite surreal, but we actually were following this story and thought we've got to call D2E7 Eric because he's a fantastic sportsman, but also this idea that if we follow Eric long enough, we might get a few sardines. So it was quite a time for Manchester United as well as for HumiRa. So we started out talking about the commercial success of HumiRa, and obviously that's important. But I think probably one of the best days that we had after launch of HumiRa was when a patient actually came in to talk to us. So this lady Vanessa came, who was a rheumatoid arthritis patient. And it's sort of going back to what Ian said, that really it's all about making a difference to patients, and it doesn't really matter what technology you use. When you hear a patient stand up and talk about her story of really understanding what a debilitating disease rheumatoid arthritis can be in a relatively young woman, that she couldn't turn on a shower, she couldn't open a jar of jam, and she talked about the benefits that anti-TNF therapies had brought to her. And that was a real light bulb moment for us, because I think up until then we'd been very focused on the antibody engineering, the scientific academic sort of mechanisms of what we were doing. And I think this really made things very real for us, and that was a turning point. And I think keeping that patient perspective in everything that you do is vital, and certainly within the Medimmune organisation, which is what we're part of today, having those patient stories and bringing those back is really important, because it really does make you realise the fantastic opportunity that we as a community have to deliver benefits. So that's the discovery process, and just if I've got a few minutes left, just to sort of talk a little bit about rheumatoid arthritis today and the treatments. And rheumatologists have had a really evolving remit over the past 20 to 25 years, and their role has really mirrored the development in treatment, and biologics have provided a lot of opportunity in terms of innovation and efficacy in combination with small molecules as well. So certainly methotrexate's an incredibly valuable tool for rheumatologists as well. But there are more and more better treatment options now to empower rheumatologists to act more decisively and go in earlier to really prevent this joint damage. So if you look at the treatment paradigm back in 1998, the starting point was non-steroidal anti-inflammatories, which were taken for a reasonable long length of time. Hydroxyquinone, methotrexate combinations were really the tools that were available, whereas nowadays the treatment regimes are much more transforming in terms of quality of life, and there's earlier and more aggressive interventions. So you still go for the NSAIDs, methotrexate, but then you have this series of TNFs that are available that can hopefully deliver significant benefits. And if you look at the biologics marketplace, we've mentioned Entaraset, we've mentioned infliximab, HumiRa. We also have the slightly more recently approved anti-TNFs, Simply and Cimzia. And we're also looking at other mechanisms, other biological interventions, such as the T-cell co-stimulatory modulators, CTLA-4 fusions, CD20 B-cell axes, and other cytokine inhibitions. So there's a lot of opportunity, and there's still scope for innovation in rheumatoid arthritis treatment. So it's nice to see how this has evolved over the period of the past 25 years. So just two more slides briefly to summarise in terms of what more is there to come. And if you look at approvals in terms of therapies by source from this is cumulative 2009. If you look at where we were in 2009, there were a significant number of humanised antibodies out there, only 3 human. Over the past 5 years, that picture shifted. So we now have, by my reckoning, and apologies to Jan, who did a great summary of the position in terms of approvals last night. I think, by my reckoning, we have 10 fully human antibodies. So that picture's changing, moving away from the chimeric and the humanised to fully human. And as Ian said, transgenic mice technologies have delivered a significant number of those. Phage technologies have delivered delivered HumiRa, Benlysta, Abthrax, and again, as Ian mentioned, those were developed in very nice collaborative environments with HGS and obviously HumiRa with Noel and our colleagues at Abbott. Moving forward, if you look at the Medimmune pipeline today, as Ian said, it's very much technology independent. So we have around about 30 compounds in clinical development, and I would say around 60% of those are phage derived, and we're starting to see some of those phage derived compounds moving into late stage phase 2 and phase 3. So we have a significant number of phage compounds coming through, but we're also building a pipeline which combines the various technologies. We've got transgenic mouse antibodies in there, we've got bispecifics, we've got all sorts of things. And I think we need to get away from this view of technology as a driver and do the right thing for the biological pathway that we're targeting. So I think I am presenting almost as the ghost of Christmas past, but I think hopefully in 5 or 10 years' time I'll be here as the ghost of Christmas future as well, to be able to tell more stories of what phage has delivered, and I think there are a lot more excellent compounds to come. So I realise I'm probably getting short on time, so just to acknowledge, and before I sort of go into the names here, making a drug like HumiRa is a massive undertaking, and huge numbers of people, not just within CAT, but at the LNB, at NOL, at BASF, and then moving that compound through into development, which wasn't our story, that's the story that BASF could tell, and now Abbott and AbbVie, huge numbers of people involved. I just sort of pull out the core team members who worked on HumiRa at CAT. Debbie, Henny, Brian, Andy, Tris, and Jane Wilton. But Kevin Johnson, who was our head of research at the time, was very instrumental in driving this project and really providing the sense of urgency to deliver. And if you look at the timelines we talked about there, we started work in earnest in '93, and we'd made Eric by February '95. So it was a pretty tight timeline given where we were with the technology. Dave and Greg Winter, who was a great advisor and supporter, and our colleagues at NOL BASF, particularly Bob Kamen and Jokan, who were fantastic in terms of driving this. And just, I also would like to say that Mike Neuberger was one of the scientific advisory group members of in the early days of Cambridge antibody technology, and I'd like to add my personal tribute to the contribution that Mike Neuberger actually made to the field of antibody engineering. He was a fantastic advisor, a really warm and very professional, very thoughtful person, and we got a huge amount out of having Mike on our scientific advisory board as well. So I'd just like to acknowledge him, and thank you for your attention.
Oh, okay, so Jane, brilliant, excellent. Kept to time, always good. Ahead of time. Questions for Jane?
So Jane, did you use this guided selection for any future project, or was it always, once you got the bigger libraries, that you always started from a naive library? I was wondering whether this has been used for other.
So we didn't use it for any other projects in the end, mainly because by that stage we actually combined the repertoires that we created to enable the guided selection into the large naive library. So it was around about 1994 where we were starting to be convinced that the large naive libraries were up to the task, and we sort of gradually increased the size and diversity. And I think also the nature of our business model meant that because we had the libraries, we tended not to use mouse monoclonals as our starting point. So we just went in with the large libraries and started doing naive selections right from the word go.
Because there was actually then becoming more work, because then you would have to make a new library every time.
Yes, absolutely. Yeah, and actually, one of the real issues we had in the beginning was actually cloning out the mouse single chain in the first place, which actually took quite a few attempts to sort of get that right. So. There's no reason why we couldn't have used guided selection. It worked very nicely. It's just that it didn't really make sense from a business perspective to do that.
Okay.
Yeah.
They were the days when cloning a murine antibody took time. It was hard. On all those primers, we were going out for ages and ages and trying to get the PCR right and get a fragment. And you never got a fragment. You got two fragments. You got half a fragment. They were the days. Anyway, right. Yeah. Who else? So Jane, it's a 20-year journey, right?
Yeah.
From there to now.
Yeah.
Was there a moment where you thought, actually, a eureka moment, or actually was it just a whole series of sort of steps in the right direction to where we are today? I mean, you don't develop the world's biggest drug, which is what it will be this year, by kind of accident. And yet, I'm trying to work out whether there was a real aha moment where you went, oh, blimey, we've kind of you've done it, kind of thing, or not.
I think probably the biggest single step forward is being able to make the CDR3 mutated libraries, and again, sort of going back to the prime designs and making those libraries efficient. That was probably the biggest challenge that we had, and that was the biggest single step. And actually, Tris Vaughan did a fantastic job of resolving a lot of that. But I think there wasn't really any big aha moment. What there was a lot of constant troubleshooting, very, very intense troubleshooting, and that's why sort of we have the sort of team members here, but everyone was peer reviewing what we were doing on a weekly basis, and Greg was coming down on a weekly basis and trying to work out how we could get this thing to move faster or how we could get the affinity higher. I mean, we started out with the light chain shuffling because that was easy to do. And that probably, if we'd gone in and done some serious mutagenesis earlier on the CDRs, we probably would have got there a little bit quicker. So it was a combination of the tools that we had available. But the big thing was the team attitude, actually, and that everyone understood what the importance of this project, because this was the biggest agreement. This was, if this failed, then the business would have failed. So we really had to work hard to drive it.
It was kind of important.
It was kind of important, yeah. But it was really good fun.
It's good. It's good.
Yeah.
Just going to show good fun can make big money.
Yeah.
There's a moral. Look, our very own Laurent Jespers.
Okay, great summary, Jane. So you started as the underdog, obviously Remicade, and then we're already there. So what is in your sense that made it to become the best-selling drug?
So why do you think it was? Well, I think there are various advantages in terms of administration, so self-administration, I think, which was all driven through the Knoll Abbott axis. That was a really good decision to make early in the development. So 2-week self-administered compared to infliximab, which is IV, and not having, so the other big competitor was the receptor fusion, which had marginally less efficacy. So I think that the route of, in terms of those early trials, it looked like the route of administration and having a whole antibody actually seemed to give us a benefit. And all the TNFs sell very well, so it's a little bit of luck. But I think you have to have a differentiation strategy, and so this was the first Biosuperior we made, but we didn't really think of it as a Biosuperior in those days. We just thought, right, well, how can we give something that's not going to have minimal hopefully anti-drug reactions, and have a sensible route of administration and PK/PD.
Okay, there are no further questions. Can we thank Jane again? Thank you, Jane. Good job.